Hey everyone, my name is Alex Yonk. I'm a fourth year neuroscience PhD candidate at Rutgers University. Currently, I work in the lab of Dr. David Margolis, and my research interests are sensory motor integration and neurodegenerative diseases. So you may be asking yourself, what is sensory motor integration? Aside from being two very difficult words to pronounce, it's the combination of sensory, so for instance, touch, taste, hearing, smell, and motor, in this case, it can be an arm movement, the combination of those two pieces of information to perform certain actions. So imagine this is you, a blonde haired young boy walking down the street, and all of a sudden you see a $100 bill lying on the ground. You really want to pick up that $100 bill, don't you? But how do you go about the process of picking up that $100 bill? Well, let's break it down. What do you need to sense, and how do you need to move to pick up that $100 bill? So a couple of actions that you probably need to take are you need to see the bill on the ground, you need to approach the bill, you need to bend down or bend over to pick it up, you need to move your arm towards it, you need to feel the bill with your fingertips, you need to pinch it in between your fingers, and you need to maintain your grip on the bill as you continue to stand back up and maintain your balance throughout the entire process. Of course, there's going to be a couple more. I'm sure you can think of some. But all of this leads to your expected action, picking up the $100 bill. But there's a side of it that you don't really realize. What are you not doing when picking up the $100 bill? A couple of actions may be reaching to a different area. You're focusing on picking up the bill. You're not reaching to the left or to the right. Normally, at least, you won't be wobbling when you're bending down or standing up. You're not dropping the bill after you pinch it. So, in essence, there are two folds of sensory motor integration. On one hand, you're using the information from a bunch of different senses to perform wanted actions. On the other hand, you're also using that same information to stop unwanted actions. But why do we as neuroscientists care about sensory motor integration? Great question. So there is a very long word again called neurodegenerative diseases. Never heard of them before? I'm sure you've heard of at least a few of them. So these are diseases that result in either the progressive damage or death of brain cells, also known as neurons. They're split into two categories, one of which is dementias, which relates to mental functioning, so you can think of memory and mood. And the most common of these is Alzheimer's. And then the other one is ataxias, which is what my work is more geared towards. So with sensory motor and ataxias, this process is known to be impaired in a wide variety of these debilitating ataxic diseases, such as Huntington's disease, dystonia, Tourette syndrome, and the one that my research most closely resembles, which is Parkinson's disease. So how does my research fit into this? Well, I'll be completely honest. We as neuroscientists don't know how the brain specifically makes decisions. Once we learn about this, we can see if there's differences between normal and ataxic brains. And once we find differences, we can target them for treatment. To this end, my personal research uses mice to see how they can make decisions with their whiskers. Then I try and change the activity of certain brain areas using a technique called optogenetics to influence their decision-making behavior. So this is a really cool GIF posted by Josh Jennings, who shows that although it's a very complicated technique, it's very simple to understand. So all we're doing in this video is we're showing a mouse that is exploring in an environment with food there. And when they change the activity of the brain cells, so in this case, turning on the blue light, the mouse begins to eat the food. When the blue light is off, as in the beginning of the video, the mouse is just exploring its environment. So what I'm doing is I'm using two different colors of light to change the brain cell activities in the same area to see how it changes their decision making. So blue light means I'm turning cells on, yellow light means I'm turning cells off. And then afterwards, I need to check if I was